that's in fact, that is going to be my focus. Okay, now I've started the recording type of thing. So we'll go ahead with this. And now this is, you know, I did the class on human evolution and origin of life. So this is life. Cancer is part of life, basically. So, and this is a classic statement that was given by a Russian biologist, and nothing in biology makes more sense except in light of evolution. So one can say that cancer is an evolutionary, concept that we are living with, and okay, and death, okay, and up until now, unfortunately, the main cause of death is people. I did a class on aging a while back, in the science of aging, and actually there is even an idea that aging should be classified as disease, so it can be treated as such, and even more importantly, so the doctors can bill you for treating for aging. No, I'm just joking, but no, but actually, so it would increase the funding, and type of thing, because that is the cause. And actually, World Health Organization has classified night shift work, as a risk factor for aging, basically. I mean, it is actually the truth, and even just like so, and even like, for example, people who are like working with airlines, and their circadian clocks are disturbed, so that just changes the aging. But until that does that, we're stuck with the causes for death, like which are okay. And these are the traditional causes. This is an old slide, but it still makes sense, So until aging is classified, you have to use this table as a cause of death, And worldwide, you can see that, ischemic heart disease and strokes are way up there. And perhaps that may be due to poor treatment of blood pressure in some of the developing countries and even in other areas, While in the United States, the malignant neoplasm is right up there as number 2, Even with all the COVID was number 3 even in 2020 during the first wave. So, and there are some, I didn't write down the reference, but at least in few years, it may be that malignancy may become number 1 and ischemic heart disease may drop down with better treatment with statins and other stuff, so that things may change, So, and so these are pretty scary statistics, type of thing. Okay, and this is now just to give you a historical perspective. It's amazing that the government website, your tax dollars at work, cancer.gov, has an amazing wealth of information, and I just put this slide in for the first time, this morning, because this traces I'll throw in from time to time during the talk, that these are the milestone in cancer research and discovery, and you can read it yourself. And this is like a who is who of, I'll have individual slides about all of this, but just to show some of the well-known names, and I'll, type of thing, that this goes down to Sir Percival Pott, who proposed chimney sweepers. The little boys who are 4 and 6 years old were tiny enough that they had to sweep the chimneys, and they got testicular cancer. And it took about 50 to 70 years before the grandees of the parliament to pass a law. Says, okay, I mean, a few little boys died, so it's no big deal, So they dragged their feet for a while, even after the proposal to pass the laws, and then we'll talk about individually as go around type of thing. But I think this will be a nice list, and for those of you who really want to look at the thing, there are individual descriptions of all these items at this website. So this is a clickable link, by the way. Okay, when you get the slide, you can even print it, this is from the cancer.gov website. Okay, but now all of us who are old enough to remember this song by the Stones, Mother's Little Helper.
Yes.
And I just threw in Mother Nature's Little Helper. Okay. And so that is, and you can say this is the second line of the Rolling Stones song was, it's a drag getting old, Okay. Okay. and they were spot on, and I think, now, and obviously, I think some of them, even with their lifestyle, I think they're still around. So this slide actually shows that this is from a well-known cancer biologist researcher, that even though, if you can see I think, okay, let me see if I can zoom in on to, oh, no, so sorry, I'm dealing with only one monitor. So, but you can see the slide, that in the cancer, even though most of the cancers, happily for us, starts mostly after age 60, with a couple of exceptions, the two exceptions being testicular cancer that occurs at a young age, even a 30-year-old can get testicular cancer. And that may be because it's a germline cancer, so to speak, type of thing. And fortunately, again, Mother Nature is kind, that the ovarian cancer does not start at a younger age, there are some, but testicular cancers can strike. And some other rare tumors in childhood called retinoblastoma. That is really an outlier that, it peaks at age 2 or 3. And by the time the child is 10 years old, it's almost, decrease the incidence. So there are two outlier cancers, okay. And the testicular cancer, first peak is at 25, second is around in the 50s, so, and so that may be, so, but, and again, you can see how the chances are. And even with smoking, even if you smoke, the cancer, it takes about 30, 40 years of smoking for the cancer to show up, type of thing. So, and this may be, again, you think in a broad sense, you would ask that nature has programmed us that at least, she won't kill you until you have had left a copy of your genes. Okay. So, after that, you've served your purpose. Okay, then it's okay. You've done your job. It's okay that if you check out, So there may be some this thing. And I think Peter knows that there's another equally interesting book that I shall share with you, type of thing, it's called Rebel Cell. It shows the relationship and the complex nature of evolution, genetics, and the other features of our tissue that, so I'll talk at more length as we go along. Now, I think all of you are old enough to remember this, that this was 1969 ad that was taken out in the Washington Post and New York Times, okay? And this was done by a very active group that was advocating increased funding. And it's an interesting story. There was the Lasker Foundation, and that was started by a woman named Mary Lasker, Lasker, and she was ahead of her time. She was born in 1900, had gone to Radcliffe, and was obviously savvy enough, married a rich executive, and had afterwards had devoted the cause to, and her husband had died of lung cancer, by the way. And so she had devoted her energy to funding cancer research. And she single-handedly succeeded in, swaying public opinion, along with the first man who started chemotherapy at the well-known Dana-Farber Institute, that was named after Dr. Sidney Farber in Boston. So this was the start of Nixon's campaign, and the funding went up from 1940s, late 40s, from $10 million to almost like $500 million, and that was the start of the big research that was carried out under the auspices of NIH and National Cancer Institute. And again, has that been successful or not? The answer is it is complicated. I have a separate set of slides, which is called the success story, and so I will share that with you as we move right along. Yes, we have made, we have a 50% chance of making through, and we have made tremendous progress in hematological malignancies, but still some of the big ones are tough nuts to crack, especially lung cancer and pancreatic cancer, and even advanced colon cancer. And obviously, now finally we are realizing that, I don't know why it is that we just always want to declare war on everything, instead of trying to live with it, type of thing. And there are some parts that it does make sense to like you live with diabetes, or you live with asthma, perhaps, so there is some evolutionary thinking that is being done right here in this town, and I'll share that with you. And again, so this is the latest episode of Cancer Moonshot that Obama and had proposed, and Biden has reaffirmed just as recently as this year. But this time, the goals are more reasonable: to diagnose cancer sooner, to prevent it, to address the inequalities, and to address more childhood cancers, the rare tumors, and try to find the right treatment for right patients. At the same time, give support to patients, caregivers, and survivors. Because as we all know, cancer just doesn't affect one person, okay, type of thing, but it affects the whole family, type of thing. And it's like, the process of grieving and a lot of psychosocial aspects are equally important. And as physicians, we are probably very guilty of neglecting that, type thing. Now, again, the Brits tend to follow what US does quite often. And on the same day, the British Health Minister, Sajid Javid, had announced also a UK cancer moonshot on the same day, by the way, just for the Brits in the audience, So how that will fly, that time will tell. Okay. Now, cancer is, we all think, oh, it's our lifestyle and the hectic lifestyle and cigarette smoking and the pollution and all the chemicals that the nasty chemical companies are adding are causing cancer. But wait a minute, that's not true. Okay, cancer is about as old as human beings, or even older. Okay, Egyptian papyrus named Edwin Smith papyrus. Now that's obviously a very Egyptian name, called Edwin Smith. Okay, and that showed, so that had mention of cancer. The description was there, obviously not the diagnosis, but it describes very vividly what could be classified as breast cancer. And going back even further, this is almost blows your mind. 1.7 million year old dinosaur bone, and also the from the cave in South Africa, and the earliest humans, there's Homo nabilis, that they were able to find the bony evidence of cancer, and the dinosaur skeletons from 76 million years ago. This poor dinosaur had cancer, so it's not a new disease by any means, and and also then as recent now, the wonders of DNA is that there was during the Renaissance age, there was an emperor Ferrante of Aragon. After 500 years, his skeleton was correctly diagnosed as having cancer. Okay. Now, then cancer is in all sorts of animal species, and I may have a slide later on for it. Now, somewhere there was an exception that sharks don't get cancer. Okay, and there was a book written in 1992 that sharks don't get cancer, and that just created a big this thing on killing sharks and using shark cartilages, because shark skeleton is made of cartilage, and which does not have blood supply, and perhaps there may be some biological reason that's why they don't get cancer. And when you look at the evolutionary thing, that placental mammals have more cancer, and part of the trouble may be placenta, because even though you love your babies and obviously we are all trying to save the fetus, but placenta is a very invasive, it's like a foreign invasion, because half the genes are not yours. So that's it has developed, mother's immunity is toned down, so to speak. So, and that may be the reasons why that as mammals we are more prior to, and then again, physicists have this awful knack of trying to invade other terms. For example, we talked about Erwin Schrödinger's book, What is Life, where he was a nuclear physicist and dabbled into biology, and that was actually worked out very well for biologists because it changed their thinking. And so some of in 2011, a paper was written by a couple of physicists that cancer is something called atavism, means going back to on the evolutionary scale, like sometimes you may have heard about babies being born with a tail, and that's So it's a vestigial organ that's left. So it's like we are regressing into the past, so to speak. But biologists did not think kindly to that idea. But there may be something to that, that is an evolutionary thing, okay, that somehow along the line, some of the cells decided what's in it for me type of thing, and decide to behave in a selfish manner and try to grab the resources and do not follow the real rules of the multicellular society, okay? Because if you're living in a society, you are expected to behave with certain rules. But there could be some people who are cheats, and they want to grab onto all the resources, and, say, not pay their taxes, or, just try to look out for themselves, so to speak. And sometimes they do succeed, and even go on to become presidents, So sometimes, so that may be that some cells do behave, and that is the concept of evolution, that some cells become rebels, and that may be the reason why, they just break all the rules and do not behave and just, hide secretly. And there is a lot of evidence to that, that is being thought about a role of evolution into cancer. Okay, enough said on that. Now the cancer is in all the other things, like even trees. Some bacteria can invade the trees and grab the resources and can form tumors. So this is the tree gall tumor. And again, this is the example of the chimney sweep, that was associated with testicular cancer. And you can see this poor little child, who is, just because of his size, and the aromatic hydrocarbons were obviously the culprit, type of thing. And this was, there was no scientific proof for it, but it was just an observational study, basically. Okay. And then, but later on, it took about another 100 or so years. In 1915, Japanese scientists actually painted coal tar on rabbits' ears. And sure enough, the rabbit ears developed skin cancer. But obviously, he was Japanese, and his research fell on dead ears, type of thing. And it took until long time to prove the association of hydrocarbons, until later on. Okay, so this was the first use of an animal model actually to prove that the substance is carcinogen. Okay. Okay. Now this is the Greek word for cancer, which is basically crab. I think those of you who have the classical schooling can recognize this. I just copied and pasted it from Google. Okay, so this is the standard thought that is in all the textbooks that you'll see. What is cancer? Cancer is when a cell picks up genetic mutation and multiplies out of control. And for the most part, it is true, but biology is not physics. It's messy, and it's not just genes, because we've said, like we read selfish gene in the science grade book decision class. And it's a lot more complicated than that, it's biology is, so there's a lot of effect of environment. And as we have seen, it can say just about all the classes that I've done, have some role. For example, lack of sleep, and gut microbes. We talked about it just last week, that the microbes that are friendly and help you make vitamins and help your digestion can again mess up your immune system, and again can weaken the immune system, and later on leaving a window wide open for getting cancer and tumors. So this is, so it's just so we have trillions of cells in our body, and they really need to follow the rules and not overpopulate. The mess should be cleaned up, and they should not, when their time is up, they should need a quiet harakiri and commit apoptosis. And so, but that doesn't happen. So when some cells go out of whack, that's when cancer starts. And the same thing happens with chromosomes. That chromosomes, we are not always this. If you look at cancer cell chromosomes, they are not all nicely arranged in 23 pairs. It almost looks like somebody, a crazy artist. It looks like a Picasso painting, basically, even worse. Type of thing. Jackson Pollock type thing. So the chromosomal, it just looks looks like a big mess, so to speak. And this is again, when you look at the evolutionary thing, that we all started as a unicellular organism, like first without nucleus, so-called prokaryotes, and then acquired nucleus and became eukaryotes. And then the evolution just took off with the availability of oxygen. And there is actually a tiny amoeba, it called dictysolium, that normal circumstances, it is unicellular, but when the food is low, it decides that, okay, we are going to cooperate with others and becomes aggregate, okay, and lives in clumps, and then it can actually sense food and try to find out where the best available food is. So we'll cooperate with other cells. But again, in this cooperation, some cells are cheaters, that they'll grab more resources and they'll manage to survive. And they'll be more friendly to the cells that are biological, genetically similar to them. So you can just see as to how even in this tiny unicellular organism, the idea of grabbing resources was already forming to something. And this is that's why I love this Kate Arney's book, that there are even in some like at the time of rut, the deers go around fighting, and while the two male bucks are fighting, there are some other smaller deers. They would just sneak in and have, and try to, mate at the same time. So they're kind of sneaky, they would try to duplicate as a gene. So it just goes on that even in societies, some either due to wit or due to cheating, they can grab the resources, can multiply, so to speak. Okay, so this is just, so we already talked about that disease is not just modern. Okay, and this is interesting that this is a painting that as early as an English physician named Dr. John Hill, as early as 1761, he had published a study on the harmful effect of snuff. Okay, basically snuff. I mean, obviously, all of you know snuff, which is inhaled through the nostril, and yet associated with like tumors of the nose, and immoderate use of snuff developed cancerous lesions which could be fatal. So he was ahead of his times, obviously. Later on, another physician in Maine had proposed correlation between lip cancer and pipe smoking. Okay, so this was, and then it just continued that somebody else had written in New England Almanac and Farmer's Friend that tens of thousands died of diseases of lung brought on by cigarette smoking. How is it possible to be otherwise? Tobacco is poison. But it took until 1964 for the Surgeon Generals to wake up. And it's a long story as to how the lobbies, Now, interestingly, and obviously this is a picture of Ulysses Grant, who was always there with his cigars, and he eventually died of throat or lip cancer type thing. So this is the famous general. So this was a picture that I found out somewhere. So that's him sitting on his chair with his cigar type thing. So this was, it's a well-established use, and the Royal Infirmary, as far back as 1859, condemned smoking led to cancers. And in the modern time, the Nazis were far ahead of the other Western countries in putting association of tobacco and cigarettes. So they would just, and they were much more health conscious, and perhaps, their leader Hitler was deathly afraid of cancer, and that may be some reason why some of the cancer researchers who are Jewish still were not kicked out, from Germany, because but again, their research, whatever research that there was considered frowned upon, and nobody wanted to pay attention to their data. And it took until 1948 to two physicians named Richard Dahl and Bradford Hill, who were the public health epidemiologists and experts, conclusively showed that yes, there was a lot higher mortality with smoking. So it took almost 200 years for the society to catch up the relationship. Okay. And so these are the years that I had, but so and now again, it's like the origin of cancer. We go back to the origin of Western medicine of Galen, Cilius, and again at that time it was considered a bad humors. It was just like with the Plague and other stuff, or bad malaria, bad air. So the same way, this was considered to be black bile that was the cause of the disease. And that was Galen's thought type of thing. So, and it was, and the same way it was related to depression, melancholy, so perhaps cancer and depression went along together. So maybe that's why he labeled it as black bile. Okay. And so this was one of the associations, but obviously that didn't. And then later on, there was an English anatomist named Hunter who thought, okay, well, maybe the lymph, which is the different type of liquid, carried cancer cells to other part of the body. He was a excellent part anatomist, and the father of English anatomical museums type of thing. And so this was, and so people had gross anatomy available, but with the more and more availability of microscope, it took until the Germans, where the father of modern pathology named Rudolf Virchow, his statement was that he observed cells, and this was his famous statement. Obviously, they all spoke in Latin at that time. Omnis cellula e cellula, means life comes from cells. So that was, and he was, and before that, some other Germans had, they were all, aware of infections at that time in pus, because remember, I think that they wanted to see something called laudable pus for healing process, for abscesses and things like that. This was before antibiotic and before antisepsis, So this was, and when they looked at some person's blood, it almost looked all full of pus. And it turned out it wasn't pus, actually. They were looking at leukemia. And Virchow, who was not just a pathologist, anatomist, a social activist, and actually believed in improving the health of the German, republic type of thing, and took on the Kaiser himself, basically, and so he had coined a lot of these terminologies that we know in pathology, and he coined the term leukemia, and then he discovered various pathological concepts, such as hypertrophy. If you do weightlifting, your muscle will become hypertrophy. It will enlarge in size, but it will not become hyperplastic. It's the number of cells will not multiply. Okay, so these are the two terminologies that those of you who took biology 101 will remember, that these are the two terminologies. And later on, the cells changes this thing, and then it becomes carcinoma. And then the various stages that one goes through, carcinoma in situ means localized. Okay, that is just the breast cancer earlier. And then it breaks through the various membranes, invades the muscle, and various stages take place. So this is one way, the general pathology, this is what we were taught, before the days of molecular biology. And I'm sure that the physicians and the nurses amongst you will remember this very well. This was the classical teaching that all of us went through in our pathology textbook. And so this is, and just a definition, just to refresh our memory, forms in the skin or tissue that internal organs such as kidneys. And now the two different types of tumors. I'm sorry. Yeah. Okay. Yeah. Okay. The carcinoma is generally the epithelial tissue that arises, from the inner lining of the internal organ. Sarcoma arises from connective tissue, which includes fat, blood vessel, and even placenta. Can develop a chorion carcinoma type of thing, osteogenic sarcoma, that type thing. So this is, so these are some of the different two categories, and the rate of growth is different, and the various other terminologies that there are.
Even 5:30 or 4 to 6 or 6.
I'm sorry.
I'd say 5:45 at the latest.
Okay.
Okay. All right. Yeah. Please, if you don't mind, mute yourself. If you're, unless you're posting a chat. Okay. So there are varieties of carcinoma. That adenocarcinomas of lung, colon, breast. And the squamous cell carcinoma, and there are other types of cells. But that's just unless you are studying for pathology, they're not that important. Our discussion, that type of thing. And so these are the classic symptoms of cancer that, a lot of times by the time you get symptoms, it may be too late for certain cancers. That just makes it very difficult to diagnose, especially things such as lung cancer. Now, obviously, the classic, I've always said the best job would be to be a dermatologist in Australia type of thing. So this is, you would never go out of business, you would never run short of work. So because all the pale Brits who settled there, and who are sun worshippers down in the tropics, would be, they're set up for one of the highest incidents of melanomas, type thing. So, and so the changes in skin features and a lump, obviously, the importance of breast cancer, difficulty swallowing, and that's, that chronic cough, weight loss, fatigue, abnormal bleeding. So these are the classic symptoms type of thing. So, but again, the symptoms appear far later, so it makes it very difficult to diagnose. Okay, and this is again, so there are now this is actually that the technologies, running ahead of it. That there are various apps, and some are you pay for it. There are other ones that are through University of Michigan, and they're at least reasonable in trying to assess whether the skin cancer is something to be worried about or not. So that's where there's an app for that. It's coming. So this is one way to stay ahead of this thing. So, and again, there are various diagnostic tools that we have used, including just getting regular physical examination, various imaging technologies. Some are useful, some are not so useful. For example, annual chest X-rays, which were done as a routine when we were in training, they didn't pan out because we're still not able to make early diagnosis, because the image interpretation was difficult to interpretation, and by the time you could see it on regular chest X-ray, again, it was too late for something. So there are some other modalities that are being used. Low dose computerized scanning for regular smokers, is advocated in their various trials, and that's been fairly useful. And obviously, all of you who are Medicare age have received the test for screening for colon cancer. We talked about it when we talked last week. Okay, and again, then something happened in 1910 that a physician named Dr. Rouse was brought a chicken by somebody in Connecticut, and that chicken had a tumor, and he was able to isolate virus from that, and there was a tumor virus. It was called the Rouse sarcoma virus, because by that time, people were able to filter and isolate viruses. The knowledge was available by that time, because obviously, other diseases and vaccines were, already being utilized. So that was the start of the Rouse sarcoma virus. And he proposed, obviously, he was a good campaigner, and his word was well respected, that tumors the viruses may be cause of cancer. And again, it took another 40 years or so. A physician in Africa was able to see, this is named Burkitt, who later on became a very prominent immunologist. Sorry, I should have warned people about some of the pictures may be a little hard, to a stomach, so to speak, but again, I think all of you are self-selected, so I will, warn you as far as possible for those who have a queasy stomach. So this is called Epstein-Barr virus, and this is a tumor that occurs in young age, and this is clearly a virus, related, which is, that's very quickly it appears, and it was later on it was diagnosed as Burkitt's lymphoma. Okay, so it was and there are various other cancers that that occur in animals, especially in dogs. It can occur, that is transmitted by sex, by the way. And the poor Tasmanian devils in Australia, that the whole population is being decimated by a virus that is transmissible, by biting each other, so to speak. So so some of the viruses are, and so that became a very popular theory. And so the again, various viruses, even in smaller animals, have been diagnosed. And then again, some of the other bacteria have been also associated. Last week we talked about H. pylori as can cause gastric cancer as a cause. Then again, the chlamydia, sexually transmitted diseases, has been associated with cervical cancer. And last but not the least, the well-known association of cervical cancer with virus. And not to be outdone, even the small gallbladder with chronic salmonella infection has been associated with a rare gallbladder cancer. And a rare type of streptococcus called Streptococcus bovis has been found associated with colorectal cancer. So a few isolated bacteria and viruses have been associated. And that probably threw the people off the course, as to say viruses may be. Now, again, in developed countries, that is far less common, but in low and middle income countries, that does still play a role. Human papillomavirus, hepatitis virus I alluded to last week, Epstein-Barr virus we talked about. Okay, and again, so this is, varieties of other things, and certain personal habits are also well known, as for example, in India and other parts of South Asia, bitter nut chewing is a habit, which is a type of nut that people chew. Along with that, they add tobacco along with this nut, and so that has been associated with cancer of the palate. Okay, so there are varieties of environmental causes, and the classic textbook that I read, which was written by a Canadian pathologist during my medical school, named William Boyd. He was at Toronto General. We swim in a sea of carcinogens. So that was a poetic statement type of thing. So, and that, but and at the same time, he had this is I mean, you have to chuckle at this time. It was how out of date the books some of that were. That in the book, the textbook that was printed in late 60s or early 70s, the pleasures of smoking shall never be known to those who have not smoked. Okay, and all of you are old enough to remember, this the lucky strike, most preferred by most doctors who smoke type of thing. So it's like this. So it was, so medical profession was exactly not, up to board, and they were, playing right along. They were in the hands of the tobacco company. And this virus idea just took off, that even Life magazine had a cover that, that this may be, okay, let's see if I can, okay, cancer may be virus. So this was the 1960s cover of Life magazine. And last but not the least, there is a rabbit with a horn, so this was thought to be, this was, that there was suspected to be, Old West tale, rabbit with a horn type of thing. So this was like a tumor type of thing due to virus. Okay, and so and varieties of other cancers have been associated. There's like a special type of leukemia that has been associated called T-cell leukemia that has been, and hepatitis I already talked about. And then there are other things that we'll talk about. There are some exceptions to the rule, that type of thing as to, but, and again, there are some animals which have evolved to the point that they somehow manage to avoid cancer. For example, there is a weird looking rat called naked mole rat that just lives underground, lives for a long time, and that is somehow it does not get cancer. And the reason for that is it's developed more specific enzymes, and it, so it keeps the cancer cells in a check, and so it does not let them escape, the company, so to speak. And there are some other animals have also evolved certain mechanisms. For example, there is a special bat that has developed continuous telomeres repair. So that's how it manages to avoid cancer. So evolution has branched out. It's not a straight, one line or a laddered approach. So there have been various mechanisms that have evolved all across the animal kingdom. Okay, and we already talked about the various viruses, and then again, this was that whether they only 2 or 3 states have mandatory cervical vaccine, which is as much as 90% effective for cervical cancer in teenage girls and even in boys, and only Rhode Island, District of Columbia, Hawaii, and Virginia are the ones that mandate vaccination in 6th or 7th grade, and there was some talk about it in Florida, but I don't think it has progressed any further, and perhaps with the current climate, it may not go any further either. Okay, so this is, again, this is one of my favorite slides that, evolutionary logic of vulnerability to cancer. We already talked about this particular statement. the cancer clone progressed by natural selection. And again, so this is under selective pressure that it develops type of thing. And this is an example of this is a famous photograph of image of Charles Darwin and an isolated cancer cell, with throwing off blood vessels at passing so under electron microscopy. So the varieties of things. So because under selective pressure, because the cells which are likely to respond, we kill them by vigorous chemotherapy, and the few outliers who are hiding underneath manage to survive. And this is interesting that how the clumps that we may be, and just yesterday I reported that, that instead of all along we have been studying bacteria in a petri dish, but Princeton people have managed to grow bacteria in a three-dimensional culture. And they've shown that the bacteria that are on the inside, they're kind of, deprived of resources, and they don't grow as well, which just makes sense, like in a colony, or like remember the penguins that are in Antarctica, they take turns, getting out, being exposed to the cold. But in bacteria, their hosts are inside, they get less resources and are far less likely to grow. And there is even report that cancer cells can hide inside other cells, so to speak, to avoid, immune response. And some of the cells can actually go dormant in lung cancer. So, well, you're giving chemotherapy. Okay, well, I'm not going to start multiplying right now. I'll wait until your dose is finished. Okay. And there are even sex differences that, male and female response rate may be different. That may have to do with our innate immunity. So it's a lot more complicated, than simple genetics. And the point is that, we have the big emphasis right now on studying the genome of everything, but we're studying DNA in isolation, so to speak, of crushed cells in a test tube in vitro, as it is called, and it does not duplicate what is happening in vivo, because where the cells are surrounded by stroma, are surrounded by immune response, and so we are not studying them in their natural condition. And that's the same reason why birds in captivity will live longer, because they don't have to fend for resources. So the same way we're studying the cells outside in a test tube, we may see a different outcome. Okay, so enough commentary about that. And so this is again just to rehash this thing. It's an inevitable part of life. Cancers can act as cheats that break the rules. And mutations were thought to be rare, but no, they're very common. Because we have billions of cells and they're multiplying every day. You're replacing red blood cells, you're replacing it in 2 or 3 days, and you're replacing your stomach lining every 3 days. So again, with all these changes that are taking place, that's certainly likely that some of it, maybe, and again, so that's what perhaps explains why we haven't won the war on cancer. Okay. And so so we have made progress, but again, at the same time, we are fighting a very uphill battle. So, so it is, we already talked about how long it's been around, and some of it is something called, in a multidisciplinary approach, that is something called the theory of randomness. That by chance it just happens, so to speak. That this thing can actually go berserk and cause trouble, and so through no fault of your own, you can just end up with, and just to quote Forrest Gump, as happens. So that may be what some of the outcomes. So, but this was a very intelligent paper from some place called Royal Society, in 2014, and it explains some of this thing in a lot more generalized cancer, complex multicellularity, cheating in cooperation within neoplasms. And so that, just some of the stuff that we already talked about, and this little fancy diagram, it talks about, how the various cells go with proliferation, there is programmed cell death, okay, and it depends upon the extracellular environment, okay, and then you're supposed to have division of labor, okay, and a resource allocation. So then again, if instead of resource allocation, if some cells decide to monopolize, or decides that, okay, I do not want to be inhibited in my growth pattern, that's when cancer starts. And they manage to survive a lot longer also. So this shows the problem with the outliers, so to speak.
Call from 8150
12246.
Sorry about that. Sorry about that. So this is basically so the way. What the concept we already discussed earlier. And this again shows, this is a rather complicated diagram. It shows that nearly all the species that we know of, cancer has been reported. So this is the your biology 101, so to speak. Like even from the tiniest hydra, which is like a little tiny organisms to this thing. So and again, and it has to do with the growth of the animal. That the faster growing, even mushrooms can get cancer, trees can get cancer. A lot of small animals, we already talked about rats and tigers can get cancer. So it's well known through all the animal kingdom that we know of, Okay. And people in UK are maintaining something called the inventory of various cancer genes. They're comparing them with the older genes that were available. And so we've shown that, so there's a clearly association that has been going on about what type of cells actually mutate. And this is just the standard basic information. The second most common cause of death. This was another basic online class that I took. So these are some of their data things. It was, and again, this is, we collect a lot of number statistics, interesting, but that we already know the extent of problem that is there. Okay. That is, the numbers are nothing to sneeze at, type of thing. So this is about, a couple of thousand people, dying per day, just in this country alone, and the figures worldwide are substantial. And this is who is affected. We're talking about gender. It's obviously, we know the breast cancer and the testicular cancer, race and ethnicity. Some cancers are a lot more common in African Americans, type of thing. Okay, and we already talked about some of the diseases, which we already talked about, cystic fibrosis and other things, which are related. And the Japanese had a lot more stomach cancer earlier, type of thing. So they did a lot of original work on that. And we already talked about, the habits that are associated, and depending upon where you live, has also to do with it. And just yesterday, I read a study about from somebody from North Carolina had studied about the artificial evolution, because how the African Americans were forcefully moved from one continent to the other. And obviously, it is a stressful situation that affected their immune response, their blood pressure response, their dietary habits, and also there was an unnatural selective pressure. So that may have affected how certain cancers are a lot more common in the African American community than in otherwise. Okay, and again, so this is just basically who's, what's, and where. And so this shows the cancer screening programs have made a dramatic difference. So obviously, the more recent stress that is being emphasized about prevention, and there may be more bang for your buck rather than treating after it has occurred. So instead of focusing on sickness, we are focusing on wellness, and that may be the correct approach, as it makes sense, and that's where the people of exponential medicine and the recent moonshot cancer thought maybe. So if you do get a kit from your Medicare Advantage people, do send it in. It's worthwhile doing it. Okay, and now this is I think we still have not 2 o'clock yet, so we'll just go through a few more slides if it may. Okay, and this is basically a tumor cell, for example, and then this is, let's say the problem starts with a damaged DNA. Okay, and with the damaged DNA, there is accelerated growth, and the cell says, okay, well, I'm going to have a lot more copies of myself, okay, type of thing. And then it says, okay, I have more copies, I want more blood vessels. So this is something called recruitment of new blood vessels. And there's a fancy term for that, it's called angiogenesis. And this was scientifically proven by a cancer biologist on the East Coast named Judah Forsman. And again, his idea was not well accepted, but eventually he went on to discover not just extra tumor growth in tumors, extra blood vessel growth, but he also discovered various factors called angiogenesis factor or VEGF, VEGF, vascular endothelial growth factor, and tissue growth factor. So he actually, and that has been a target for targeted small molecular medical therapy. So he did a lot of pioneering work on the angiogenesis. And in certain parts, you want the blood vessels to grow. For example, in heart attacks, okay, you want new blood vessels to grow. On the other hand, in the case of tumor, you want to starve the tumor. So so there are medications that can be designed or tailor designed for both purposes. Okay, so then again, so there is when there is an increased blood supply, there is a chronic chaotic blood vessel structure, and some of the blood vessels get compressed and some can grow new. Okay, and so then again, there's internal pressure that changes. So internal cellular milieu changes dramatically. Okay, and then again, so there is a lack of oxygen that develops, type of thing, and the cell gets into something called anaerobic metabolism, and that again causes further genetic changes. Okay, and then again it causes the metastatic spread. So how this whole cascade develops, type of thing. And obviously, when we study just the DNA, we are missing out a lot of these factors. And again, this cell does not live by itself. It is surrounded by other stroma, fibroblast, and other tissues. So it's it's a lot more complicated. And don't forget, we are everybody's individual immune response is different. Okay, and the cancer cells are smart. They secrete certain chemicals to hide themselves from immune response. Okay, and that has been the new vogue of immune checkpoint therapy, because cancer cells have evolved mechanism to hide from immune cells, T cells. Okay, and that's where and we'll get to that when we talk at the end of the therapy type session. Okay, all right, a couple of, and again, yeah, this was the last point, deactivation of immune response. That. So it's a very complicated thing. So that just brings us to the second milestone. So let's see, what do we have? We already talked about the cancer in rabbit, and then last but not least, in 1928, this was one of those. We talked about eugenics at one time, where people from other parts other than Western Europe were considered not a desirable immigrants, but this was a Greek physician who did manage to enter United States because he was, he was qualified and he was a pathologist, and he discovered, while he was studying the menstrual cycle of guinea pigs at Columbia University, then he saw changes in the cells, and he decided to look at human beings. And this is an interesting story, that he must have a very, very, agreeable wife, that he did pap smear on his wife almost every day, and she could see changes. And then he went on to study that he could see changes, into the cell lining and the secretions. And that's one of the reasons where he proposed that, yes, this is one way of diagnosing cervical cancer. And at the same time, the established gynecologists and obstetricians, but why won't you just do a biopsy? This is such a, it is such more reliable approach. So he struggled for almost 20 years for his idea to get accepted. And his point was not only just diagnosing cancer itself, but a precancerous lesion can be diagnosed. And finally, it took until 1948 where he was able to convince the who is who of medical establishment to sponsor a study. And in Shelby County, Tennessee, they did a study on thousands of women, and they were able to not only diagnose early cancer, but also precancerous lesion. And that was the start of George Papanopoulos Fellow. And that's the start of the pap smear, and that changed, the approach of, and then other advances were taking place at the same time, that we'll talk a little bit about the surgery that was done by the great man at Johns Hopkins Hospital named William Halsted, who is a character by himself. And then the National Cancer Institute was established in 1937. And then various, I think I'll talk a little bit more after the break about the hormonal therapy. And it, by the time we already talked about the Bradfield Hill and Dahl study. Okay, so we're approaching 2 o'clock. I'll pause my recording, and then we'll start in about 5 minutes. Resume recording. All right. And I think, I'm getting a little slower, but I think that's all right. I think the stuff is okay. And this is just talks about the mutations. These are the two mutations we talked about. Somatic means in the cells that you have all over your body, and the germline is, your ovarian and testicular. So that is the big thing that we'll talk more about when we talk about. I think at least, some of 6 or 7 of you will have signed up for the genes class. So we'll talk more about the Chinese experiment of germline modification type of thing. And so these are the mutation. This was interesting that I already alluded to, that people took when people who have redundant eyelid or have plastic surgery for eyelid, they examined the cells and they could see that this was mutations galore, the whole tissue. But then somebody had arguments, so, well, I mean, it's exposed to sunlight, so what do you expect, mutations there? But again, when they're doing endoscopy for hiatus hernia, even the esophageal tissue in younger people had mutations. So you can see, well, you have so many mutations, but still we are not all dying of cancer. We wait. So there is something happens as you get older. So aging may be culprit. Well, finally the mutations accumulate, and then that's when eventually cancer appears. So that may be more to an evolutionary advantage type of thing, so there may be more to it than just mutation alone type of thing. And we already alluded to varieties of other factors. And these are just some of the examples, some of the
call mutagens. But before you go out and pick at the manufacturers, it's important to know that most of these mutagens are completely natural. There are far more in our foods than industry adds to the environment. Some of these, for example, plants make to protect themselves against predators. Even water can split bases off the DNA chain, which on rare occasions may be repaired erroneously. But each cell is thought to undergo 10,000 of these events a day. So we have many pathways that repair DNA damage, and the nucleus is a scene of active DNA damage, surveillance, and repair. If DNA is mutated in a cell that does not divide, like most of the neurons in the adult brain or in adult muscle, then the chance of a tumor is low. You could say that the brakes are very strong in these cells. But if a cell can and does divide before it can repair a mutation, then there are 2 mutated cells and increasing chances of making a tumor. Some estimates are that about 1 cell per hour in the human body undergoes the kind of mutation that could lead to cancer. But nobody gets cancer at that rate. So it is generally thought that something like this happens. A mutation in a growth gene gives one cell, and then its daughters, a slight growth advantage over the other cells nearby, so a small clone develops, a clone being all the daughters of a single starting cell. If the growth were steady, say with a cell cycle time of 12 hours, then 1 day later there would be 4 cells, 2 days later 16, and on day 20 the tumor would weigh a kilogram, over 2 pounds. That doesn't happen. As we saw, lung cancer takes about 20 years to develop from the time someone begins to soak their lungs in mutagens. We think this happens. Sometimes in the development of the first clone, something happens that eliminates it. It could be that there is no room in the tiny microenvironment, or not enough nutrients, or that the immune system recognizes the clone as abnormal. But suppose one cell has acquired a second mutation that gives it a bit more growth potential. The subclone could begin to grow. This process could go on repeatedly until all the cells die out, the most common outcome we guess, or enough mutations accumulate that the body's defenses are no longer able to cope. And this could take years, maybe 20 years. Here is a sequence of mutations that has been identified in colon cancer. The exact sequence of accumulating mutations will vary from person to person, and even from subclone to subclone within a single growing tumor. The early oncogene mutations are called drivers because they make the cell divide. Later mutations are often in tumor suppressor genes. They make the cell stop growing, and if they're inactivated or deleted, the brakes are off. Other mutations affect several pathways, but many are in the DNA repair apparatus, so it gets less and less likely that the cell will revert to normal. By the time a tumor comes to medical attention, it will have an average of 30 to 70 mutations. Lung tumors associated with smoking typically have hundreds. Apoptosis is programmed cell death. Any cell can activate this suicide program if it senses that it is too damaged to be safe, so tumor cells try to do it. Here we see a tumor cell undergo apoptosis, just like the target cells of killer T cells do. Successful tumors, it seems, have, in addition to growth-promoting mutations, mutations that inhibit their ability to commit suicide in response to the metabolic chaos within them.
Okay, so he explains a lot of the slides that I was going to go through type of things. So I think that may say was some
Call mutant.
So, and this is again, this is kind of a scary slide, which I already described. This is like a normal chromosomes. They're all when they're painted with colors to make them visible. These are X and Y. The Y chromosome is very tiny type of thing. And as you can see, this is like the haywire thing of a breast cancer tumor. You can just see there are all sorts of triplicates and duplicates and varieties of one portion has been cut off and joined with other. And there's a special name for it. It's called chromothripsis, okay, which, and so this is called basically chromosomal catastrophe type of thing. And it's in geneticists use a term called ploidy. And so this is, basically, so this shows that not only just at the genetic level, but also the chromosomes themselves can also be dramatically abnormal type of thing. So this is, again, proof of the pudding lies that it's a genetic disease per se, But again, we have to reemphasize not working in isolation. And this was, and until now, so and a lot of the biologists that had to be much less organized, So back in 1999, a couple of cancer biologists named Doug Hanahan and Weinberg, put together an article, something called the, because a lot of people say, well, things are not just chaotic, so they put in a paper called The Hallmarks of Cancer, a framework to understand cancer biology. So this is, this is the same thing that we already talked about, in a nice organized manner that he put together, that self-sufficiency, okay, and then evading the death, apoptosis, limitless replication, angiogenesis, and so these are the varieties of factors that he proposed, and this is considered the classic hallmark of cancer. This was proposed in 1999, and that, but and somehow that paper, what became one of the most heavily cited papers in modern biology, that had about 36,000 citations, And then they, with some of the more newer information, they revised the article, added some emerging hallmarks, which are called enabling characteristics, that they said, deregulation of cellular energetics means the energy metabolism is or goes wacky, genetic instability, okay, and then tumor promoting inflammation, the role of inflammation was emphasized, type of thing, avoiding immune detection. Okay, so these are the enabling characteristics. So again, this again becomes popular. And this paper has been cited about 50,000 times in varieties of journals all over the world. Type of thing. So this is, the way of. Okay, so we talked about, how the varieties of factors at the cellular and microscopic level come together to cause tumor. Okay, and so this is, again, this is why you have to love Wikipedia. These are the big fancy words: self-sufficiency in growth signal. That means accelerator pedal gets stuck. Insensitivity to anti-growth signals, brakes don't work. Evading apoptosis, won't die when the body normally tells you. Okay. Limitless replicative potential, infinite generations of descendants. Sustain angiogenesis, tell the body to give it blood. Okay, tissue invasion, migrating and spreading to other organs. So it just explains in plain English what the cell decides to do, type of thing. So I think that's, so this is, again, I think this is, we already saw this slide earlier, type of thing, but I think I should have eliminated that. So this is, again, they put the earlier slide into more context as to how the newer mechanisms, so they've imposed the second circle around it, how the activity invasion, evading immunity, sustained proliferative signal. Okay, so, as to how the evolutionary concept has been incorporated with the hallmark, as to how each of them ties in. So, the English researchers have done a great job type of thing. So, again, and I've already talked about enough about the other organisms, so, and this is basically, so more details about how the cell cycle takes place, at the various levels. So it interferes with the cell cycle basically. So, and the video was very, very succinct, and he couldn't have said it better. He talks about the two classes of genes, which are proto-oncogenes, means that ones that cause cancer, and the second part, the tumor suppressor genes. So that is a complex interplay of these two that takes place in the various cell cycle. Cell goes through various mitosis, this very small part, then the various phases during the cell growth, replication. Okay, so at various stages, these genes play a role in either causing accelerated growth or suppressing the tumor. So these are the two mechanisms, that play the checks and balances. And so it's when this process becomes dysregulated, that's when the cells becomes transformed and goes haywire. And again, these varieties of genes have been subsequently isolated, starting from, remember that we talked about the Rous sarcoma virus, and that was proposed in 1910, and it took about 60 years for genetics to catch up with the tumor, that they were able to isolate the gene called SRC. So that was labeled as sarcoma gene, okay, SRC. The geneticists have the privilege, they can name it. And similarly, at the same time, there are other genes that were isolated, RAS, that's called rat sarcoma gene, okay. And the other genes, which only the biochemists would love, called MYC, which have to do with varieties of enzymes called kinases in the body. And the kinases are ubiquitous. There are, hundreds of kinases that work. And in plain English, their job is to put a phosphate group onto the proteins, type of thing, and onto the genes. So that's where their job is. And perhaps, Peter can explain it better than I can, type of thing. But okay, but I think that gets a real complicated biology. So, but these are the varieties of oncogenes have been diagnosed, isolated. And then finally, the rat sarcoma gene was isolated by two brave people called Varmus and Bishop, and they were finally awarded Nobel Prize in 1989 for their discovery in 1970s. So these were the various sarcoma genes, and so it took. And Peyton Rous himself again was the longest wait for Nobel Prize, and he got his Nobel Prize after about 70 years, when he was 80 plus years old, for this tumor that he had described in 1910. Okay, so that's the story of recognition inside. Okay, and this is interesting that varieties of things, laboratory research. This is just as a side cut, this is a mouse that has gone to Harvard. No, not really. This was a mouse that was genetically, modified, using varieties of modifications by two biologists at, so it is meant to carry a specific gene, which is activated oncogene, which is the RAS gene, which we talked about, which is under the control of mouse memory tumor virus. And so that increases the susceptibility to cancer and makes it a suitable model for research. So this routes of, by the time Harvard was given some money by DuPont, and so the invention was actually owned by DuPont. And so U.S. Patent Office, this actually granted patent to this mouse to Harvard. So this is the Harvard onco mouse. And so people who wanted to, but then the patent expired actually in 2005. So that means the onco mouse is finally free. So other people can, but the name is still registered trademark. So this is, so you can tell your grandchildren there's a mouse that has gone to Harvard. So, okay. So this is that interesting side. So how, and because otherwise originally, the genes were not, genes that are naturally occurring could not be patented. And I'll have some more to talk about in the genes class for those of you who are in that class, so it's an interesting thing. So now we've already talked about, how this, so with just for the sake of time, we won't skip too much into it, type thing. That video that was, by the way, that is from a free online class that you can actually get. It's a British website called Future Learn. Okay, so, and that has varieties of talks on subjects type of thing. And I mean, you generally, if you want a certificate, then you pay for it, but otherwise normally there, and it is time related, you can listen to the lectures, and type of thing, but they offered some very interesting classes. So that was a class on medical school that was offered by University of Colorado. So, and the presenter is obviously, you could tell from the accent, he was a Canadian physician or a researcher. He explained very nicely about what actually happens at the cellular level type of thing. So, okay, so we've already talked about it and the varieties of complex mechanisms that takes place, in altering the phenotype. Okay, and that is something, the other way I should say, by the way, that there is, this was people where he talked about how the two mutations work in conjunction, the second mutation comes and finally things take off, in a bad way, so to speak. And that was proposed by a biologist somewhere in Texas named Dr. Knudsen. This was proposed back in the early 70s of something called the two-hit hypothesis. And the ideal model for that was the tumor in the eye called retinoblastoma. And this was easier to study because we have pair of eyes. So, the children who got the cancer in one eye versus two eyes, so it's a recessive gene. So if you inherit two bad copies from both sides, then you get cancer at an earlier age, and so that's this thing. But on the other hand, if you only got one bad copy and then just through sheer bad luck you acquired mutation, then you get a one eye type of thing. So this was his proof of the pudding, that the two-hit hypothesis, and people at Mass General were finally able to isolate the retinoblastoma gene. And so Dr. Knudsen's hypothesis of so-called two-hit for cancer was proven type of thing by molecular biologists. So that's an interesting side bit as to how the idea that was proposed as a theory type of thing was proven later on by others. Okay, and this he already talked about the so-called driver mutations and passenger mutations. So it just kind of reminds you of that. I was thinking, well, the driver mutation is like the oath keepers, and the others who went wrong for the company. So this is like the drivers and the mutations, so it looks like a crowd behavior basically. So there are some people who are actually the drivers, confer the advantage to growth, and the passenger tumors who don't directly contribute, to the fitness, but they kind of go along with type of thing. So this is, that reminded you of we already talked about the things, and there are certain things, as to how role of ethnicity plays a role also, that obviously, we know that certain tumors are a lot more common in Ashkenazi Jewish groups, and as many as 1 in 40 people of Ashkenazi Jewish ancestry can carry the BRCA, or the breast cancer gene, or I should say the Angelina Jolie gene, type of thing. And there are some other genes that occur, about some rarer syndromes called von Hippel-Lindau syndrome, which causes various hemangiomas and kidney tumors that occur in a small area of Germany, and so there are some rarer diseases which are autosomal dominant, by the way. So it means you're surely likely to get it, And so, Bob, yeah, please go ahead, Bob.
Yes, I was waiting for you to mention von Hippel-Lindau disease because my son has, I guess, he's the only one in the family, but he came up with von Hippel-Lindau disease.
Yeah,
he's probably my healthiest son. It was very much of a surprise, but he's actually studied at he went to the was it Bethesda and here at Moffitt, and it was just out of the blue, but that's why I learned a little. I probably don't know much about VHL, but he turned up with it and nobody else seems to have it. Apparently, it was a mutation of these
Yeah, it's a
detector
random-
genes, suppressor genes, and he had tumors removed from his brain and from his where else kidney,
Kidneys too, yeah.
and he's
exactly.
being watched at Bethesda and Moffitt.
Yeah.
And I mean, he's healthy, he's up and around. He's, like I say, very healthy guy. My healthiest kid before anyway. And it was just all, I mean, we study about VHL for exams and stuff like that, back in school, but I'd never really come across it until it hit my
Yeah,
home
it's not that
family
common.
out of the blue. So it's interesting to hear your comments here. I was waiting for you to talk about.
Yeah, well, I'm glad they did just, I have said, thank you for sharing.
So it
your
actually
family.
exists. He has nothing to do with Germany that I know about.
Yeah, no, I figured that with the name.
Well, actually, I'm part German, but mostly.
Yeah, it would
I'm
be
not-
interesting to see the, like a whole genome analysis, type of thing to see it.
Well, apparently it's a mutation because nobody else seemed to have tested for it in the.
No, I mean, for ancestry type of thing, like the whole genome, I mean, you're in the next class, so we'll talk about it. The cost of whole genome analysis has come down dramatically, type thing. It's amazing, like how when the human genome, I'll just digress and, share it with you, like when the Human Genome Project, was like cost how many $1 million to do one genome analysis. Now I think the cost is almost down to $200 for a whole genome analysis. This is unreal, and obviously, to get the counseling might cost a little bit more, to get through the genetic counselor type thing. But this is, and I think if you're interested, there is a lady named who is a pediatric geneticist named Dr. Catherine Pope, P-O-P-E, and she offers a class on genetic counseling. She teaches, she trains genetic counselor through the USF Public Health. And about 2 years ago, I saw that her class listed, and I sent her an email, say, can I sit in your class? And she was so nice. I mean, she not only sent me, says, oh yeah, you're welcome to sit in the class. And she even sent me a special parking permit at the research building. Okay, I mean, and it was a once a week class basically. So you might want to think about sitting in on a class, because I mean, it was a small class of about 5 or 10 students, and she is a practicing pediatric geneticist. So, and if you sit in on her class, I'm sure she will not mind type, and she was, and she does actually what test should be done, how, she trains genetic counselors. So just if you have any interest, look her up,
I was so surprised when the diagnosis was made because my wife is from Korea, born in Korea, totally Korean. And we actually talked about, the chances of having any kind of genetic problems in our kids would be very, be much less limited because of, so different culturally and.
Oh, absolutely. Yeah, exactly.
And so we actually talked about that before we got married. And so it blew me away when this diagnosis was finally made, but he's doing well.
I'm glad he's found a treatment type of thing.
Like I said, he remains probably, if you look at him, he's like the healthiest of my kids by far. I don't know what happened or where that came from, but.
Okay, all right. So no, thank you. But I think I'll have a lot more. It's an interesting, scary thing about this the starting of the dominant disease. Like starting out with Huntington's disease. That was just it's a Wexler, Nancy Wexler story that goes back in the 1980s. That at least you had a treatable illness. Huntington's disease is just absolutely unreal. So she was. And she was like the pioneer for Huntington's disease. She was like the poster child, and she was, not a physician, but she just grabbed the ball and did research and collected blood samples. It's a fascinating story in genetics. I'll share that, when I talk about the genetics class. You may know about it also. Okay, so moving along. So with this, we already talked about the driver and pest mutations. I think that video was. And this is again, finally sort of talks a little bit more about it's not just genes, it's a whole pathway that takes place, so I'm not just talking about the whole, it's the whole structure as to see how, he talked about a little bit about how, at various layers, this is the gross and microscopic pathology that we talked about earlier. This is the man who is like a guru of cancer biology, Bert Vogelstein. Okay, he's a cancer researcher on the East Coast, and this is talks about the normal epithelium, hyperplastic, and then early cancer. This is the classic pathology that all of us studied. And then at each level, there are molecular activation that takes place. The chromosomal abnormalities, various enzymes that take place, various other things. And then again, at the DNA level, these are the various genes that get, he talked about in the video, he talked about the APC gene, and the KRAS is an enzyme that takes place, and then last but not the least, the guardian goes on vacation, the loss of P53. I'll have a slide coming up, type of thing. So, and then the chromosome becomes abnormal. So all this stepwise mayhem that takes place eventually leads to cancer and metastasis. So how this classical pathology, microscopic, is correlated with molecular pathology. And here is this tissue diagnosis. So this is about classical pathology to molecular pathology. So it's like it's a tour de force work, basically. So, okay. And okay, and so this is again getting down to the third stage of the progress type of thing. So we talked about and now this was interesting that in 1953, after the start of the NCI, a Chinese scientist named Dr. Ming Li, was, there's a rare tumor of placenta called chorion carcinoma that takes place, and fortunately for that, this was the early days of chemotherapy, and they were giving various toxic drugs, and fortunately that it is a marker, that it is a blood test that is available, and and the patient was actually cured, but the blood test would not become low, so he kept giving the treatment to this. I want that blood test to go down to 0, and the NIH, his superiors just thought he's doing experimentation on person, so he was actually fired and let go from NIH. But it turns out in the end he was right, that you actually have to wait until the whole tumor or the marker has been normalized before you can say put paid on the thing, because otherwise those who were semi-treated, or the tumors that came back. And finally, this was the heydays of NIH type of thing, that this was the start of chemotherapy. And initially they were going A plus A. If A doesn't work, they would add B, and this was done. But then finally people came up with the idea that yes, we have to hit them hard, with combination therapy. So the idea of combination chemotherapy, and that was not for the faint-hearted. It was like a people were working on children with hematological malignancy, and the side effects and the infections and the nausea that was there was just gut-wrenching, and there are a lot of people who were saying that, why are we doing this to the kids? Let them die peacefully. And then we'll have a story about the pioneer, Dr. Sidney Faber, that when we talked about the success story. And so this is, and then last but not the least, remember the famous report, 1964, focus on cigarette smoking, and then they, then later on, moving on, tamoxifen, which is an anti-estrogen therapy that was developed. Originally, it was promoted or synthesized as a birth control therapy, but didn't work. And finally, the UK ICI, who had synthesized, they were going to give up on it, but then somebody got the bright idea to use it for breast cancer, and the rest is history. That was one of the biggest selling drug that was for breast cancer therapy. Okay, all right. So these are some of the new progress that continued. Okay, and I think we already talked enough about the effective pathways, and this is too much of basic biology type of thing. So I think we'll skip that for the sake of time. And then last but not the least, in last few years, the science has progressed after the genome analysis, that all the tumors, there was a big project done under the auspice of NIH called the Cancer Genome Atlas that was started, and the program that is diagnosing varieties of gene in the primary cancer tumor in various cancer types was the Cancer Atlas that had been actually finished, and that has made a dramatic difference in terms of discovering varieties of pathways of various driver and passenger mutations, and people who have worked on it, Okay, and so this is again just shows the various cancer pathways in lot more details, And these are again only the biochemists would love this, the varieties of genes that we talked about, the notch gene, which has to do with the embryogenesis, okay, the sarcoma gene, the MAP kinase, the tumor growth factor genes, So again, I think I don't want to put you to sleep for it, type of thing. So this is interesting that the variety, these are again diversifying. This is something called, these are the old data, old axioms or thoughts, but they're worth sharing. There is something called the metabolism and growth. Now, this is the size of the animal and the metabolism. This was a law that was proposed back in the 30s, I think, that animals' metabolic rate scales to three-fourth the power of the animal size, So as to how you grow, that depends upon your metabolism type of thing. So that was one idea. And then later on, we would say that the larger animals do not get that much cancer. Okay. So this was, bigger the animals, they have more cells. They should mutate more, and they should elephants should drop dead with cancer, before, soon after they're born, and the whales should get cancer. But sorry, that does not happen. This is something called a Peto's paradox. Okay, this was again, I think, and the English must have a lot of free time. They come up with these ideas, type of thing. So this was Sir Richard Peto, and in 1977, he's a British epidemiologist who kept the absence of size to cancer correlation was named after Sir Richard Peto paradox. So so what is this thing? The P53, which is the so-called guardian of genome, is in the business of marshaling resources for quality control. So when it encounters the damaged DNA portion, it sort of fails to bind to another protein. Okay, it turns off a chain of event that stimulates the cell to repair any damage. If that fails, then if that doesn't work, P53 doesn't give up. So it initiates a different command that tells the cell to kill itself. Okay, so this is the so-called, this is why the P53 is called the guardian of the genome. Okay, but cancer cells again have clever defenses that they can try to turn off the P53 type thing. Now you and I have only one copy of P53, but elephants have 20 copies. So that is called copy number variation. So that's one of the reasons why they can live to be a long time and then not get cancer. Okay, and so this is P53, the guardian of the genome, and this is the mechanism: DNA damage, abnormality, lack of oxygen, protein that we talked about, cell cycle starts, apoptosis. Okay, and so this is just shows in a cartoon fashion. Okay, and for those of you who are more as nerdy as I am, and there is even a book written by, she's a very easy to read writer named Sue Armstrong. Okay, and this was a book that I think came out about a few years ago, P53, and this shows the trouble that people went through as to how they managed to find P53, that thing. So, and why is it called 53? It has to do with the kilodalton weight. Okay, and so this was the kilodalton. That's how it is called. So that's for those of you who are chemists, would know that that's how the weight of the molecule is, was reported. I think. And by the way, for the Brits in the crowd, in 1844, August 24, 1854, Dalton's funeral in Manchester was attended by thousands of people. So it was the wonder of the scientist, which was actually given a state funeral in Manchester in 1854. He was amazingly popular and a brilliant polymath scientist, Okay, but that's another story. So just okay. So this was P53. So that's called the guardian of the genomes. Okay, and again, digressing to the metabolism, so this was an interesting thing about, this was a clever German physiologist named Otto Heinrich Warburg, and he had worked on respiratory enzymes, and as a sidekick, he was also interested in tumor growth, and had proposed, as to how tumor microenvironment, how their metabolism is different, and they produce not the pyruvate but lactate. So it's a different type of metabolic process that takes place, and this has to do with the role of the mitochondria, and so he proposed that the metabolic pathway was different in the tumor, and that was the reason for tumor growth. So he emphasized the role of metabolism in now later on, obviously, that the idea fell by the wayside, type of thing, when the when the genes caught the limelight, but his ideas may be coming back, that he perhaps, and there is almost like there is a cult for Tesla in physics. There's almost like a cult for Warburg, that, yep, all these scientists are hiding what Warburg had proposed, it's what we eat in our metabolism. That's how we can cure cancer. But again, it's not as simple as that. But the role of mitochondria is becoming a lot more apparent, it's the mitochondrial disease, and there are only like 24 genes in your mitochondria, but they may be playing more role than you realize, okay, type of thing. And at the University of London, there's a scientist named Nick Lane, and he has done a lot of original work on the role of mitochondria. Okay, and we'll talk more about it in the gene class, type of thing. So this is the metabolism has a lot to do with something called signal transduction in the cell. And so there's a role of the so-called Warburg effect, maybe playing a role, in tumor metabolic processes. And the varieties of ways. So this is the classic side as to multiple factors causing. This is where the study of the omics, is, well, again, when we talk about the genome, we'll have more about, as to how, how the microbiomics, epigenome, and varieties of, how the cell, the DNA is long and it's wrapped inside the cell like a spool fitting thing. So these are called histone proteins, and how it's long. It's almost like several meters long inside the cell if you unwind it. Okay, so let's see. I think there's some more images there. Let me make this normal. Okay, and so this is the cell omics. We already talked about the epigenomics, the microbiome, the virome. Okay, and then that eventually causes the clinical presentation. So you can see how it's the whole cycle is, intricately connected and depend upon each other's progress. Okay. And so this is, again, we already talked at as to how, and this is a similar example for pancreatic cells. We already talked about the colon cancer cells, and the same example that Dr. Vogelstein gives about the normal cells, and it turns it into gradually pancreatic cells becomes, the varieties of enzymes are activated, inactivated, and the whole cascade starts. And last but not the least, don't forget the popular telomeres. And telomeres, as the classic example is saying, they're like a little thing at the end of your cells. As you get older, you start losing telomeres, okay? And that hastens the process of aging. And it's become very popular, and on the internet, you can find varieties of products that will help you make your telomeres longer, but I think a lot of that is hype. And actually, there was real science on it. And in 2009, two ladies, Dr. Blackburn and Cindy Greiner, they were the Nobel Prize winners on telomere research. Okay, and then, she has a special book on telomeres, and there's and the cancer cells again have their trick. Another trick up their sleeve is they secrete telomeres, And so they manage to avoid, aging type of thing. And that's maybe another ways how they can manage to live a lot longer type of thing. So, okay. So, and so this may be one of the reasons why there is sometimes, they get pediatric cancer type of thing. So, and so there are, again, the big problem that we can explain that a lot of the process is aging and takes years, but the two exceptions are pediatric cancers and brain tumors type of thing. So that we haven't, and that may be that because, the children's cells sometimes may behave more like rodent cells and don't require as many mutations, to turn malignant type of thing. So, okay, and that number of cell divisions are far less type of thing because they haven't had that much time to grow. And so that may be one of the reasons why for the early childhood cancer. So, but that's still a big, Achilles heel as to why to explain some of the biochemical mechanism type thing. So, and we talked about the telomeres. Okay. And again, so this you can see that the endogenous telomerase, this is experimentally proven when it is on, the tumor is aggressive in prostate, while when the tumor is off, a prostate cancer is slow growth. So this is, the telomeres have a lot to do sometimes with, with the growth and on of cancer. And prostate cancer is trickier because it is a slow growing tumor. It has far less immune activity. And so that may be one of the reasons why it's even harder to treat sometimes, when, okay. And so this is, the telomeres have a big role to play. Right? So, and again, these are some of the examples, it's a lot more complex biology than, and people are developing target mechanisms to work on this telomerase, so the varieties of drugs that are being developed and tried to work on the telomerase enzymes. And I already talked about, and we've already talked about the metabolism type of thing, that the tumor cells produce metabolic byproducts from acid type thing and ammonia, and there were different metabolic effect than the Warburg effect that we already talked about. Okay, and this is again another way of increased glucose uptake in tumors. That is, this is one way of to screen. There are varieties of diagnostic tools that we use, including CAT scan and a more expensive PET, means positive emission tomography scanning, and this is shows the glucose metabolism, of the cell. So this is actually, but these are fairly expensive tools. So the PET scanning is not used for routine staging of the tumors type of thing, but this has this is another modality for assessing tumors. Okay. All right. So I think I know, and for those of you who are more interested, they can read my notes type thing. But this is a fairly complex biochemistry, but at least and I already showed you this slide, but again a little bit more details as to age-dependent incidence of most cancers. You can see that how the process starts later type of thing. So okay, and this is again we talked about the cancer. I already showed you the slide, but I printed it again. So okay, so these are some of the more complex biochemistry side. Okay, now I think we can get onto the site-specific cancers, but I think I'm going to stop here right now and show you some of the success stories, type of thing. So we need to end things on a positive note. I'll stop this screen. Okay, and we still have a few more minutes, so I will share some of the other slides type of thing. So let me see if I can find the other two. Okay, share screen, share a portion of the screen, yes. Okay, can everybody see my slide?
Yes, I mean, there's no,
Yes.
Okay, yeah, it should say a success story.
right.
Okay, good. Yeah, okay, then we're in business. Yeah, because I had to, my other monitor doesn't work, so I'm sorry about that, but I think okay, so we'll go on and we'll show some positive studies type of thing. Okay, overview of the treatment. We already talked about, this is, this were the three modalities of treatment, that in the early days as Johns Hopkins, this was the grand man of surgery, Dr. William Halsted, he was ahead of his time. He's the first one who introduced the use of gloves and surgery, and he had pioneered the idea of, attacking cancer with, because obviously the surgeons were in their heydays, this was before even penicillin or any other antibiotic, and he had devised various operations, including radical mastectomy. So this was Halsted surgery, and this was and obviously cancer that was visible, and had a fairly good success rate, if detected early enough. But that wasn't all. Okay, and then again, in 1899, Röntgen had discovered X-ray, and even within few days, people were using it to treat cancer, especially skin cancer and other, even other cancers. So it caught on very quickly, the use of X-ray machines to treat cancer. And because people realized that it was dangerous and you could get finger burns, so the idea was easy to extrapolate to use X-ray equipment. And later on in 1905, with the discovery of radium, even internal use of cancer and using radium to treat cancer. And obviously, so that was, the early 20th century was the start of using X-ray therapy. And later on, obviously, when I think when we talked at length, when we talked about the atomic age type of thing, that when they were trying to discover the secret of the atom and the various electrons and protons, Ernest Lawrence, to get money for the development of cyclotrons on the West Coast. Okay, and he was as a sidekick, he was treating also the other patients for cancer, so that generated funds for him. But the idea of using cancer on a systematic basis, for staging, took off in 1950s, with a guy named Kaplan, who started treating Hodgkin's disease, which is a lymphoma or lymph node tumor. Okay, and with various staging and with systematic radiation therapy and regional therapy, and mental therapy. So this was, he was the pioneer for radiation therapy, especially for solid tumors. Okay, and by the way, wars and nitrogen mustard, which was used as a gas, gave the idea of chemotherapy. And some of you may recognize these two the well-known pharmacologists, okay, Goodman and Gilman. I'm sure all of us have read his textbook of pharmacology, and they were the first ones who examined the therapeutic effect of mustard gas on treating cancer, and that later on became the idea of using, toxic nitrogen mustard type derivatives for treatment. So these are the three legs of cancer therapy that came in, starting with surgery, and then X-ray, and then obviously this was the start of chemotherapy. Okay. And this is, just to refresh memory for basic cells, this is the cell metabolism. This is how your stem cells are mostly in your bone marrow, hematopoietic, and the two lines that develop the myeloid, which gives rise to the white blood cells and the red blood cells, and the lymphoid, which goes on to help you build your innate and acquired immunity, the so-called, this is, Dr. Spector, he's the guru for us, type of thing on immunity. And I think, with COVID, we've all heard enough about immunity. And so these are the varieties of cells, the eosinophil that work with your allergy and parasite, neutrophils that help you with all infections, and red blood cells and platelets, which help you with the bleeding. So this is just hematology in one slide, basically. And these are, this is one of the easiest source for stem cells. Okay, and so this is, again, so after laying the foundation for this, okay, this is the man who was the so-called grandfather of chemotherapy. He was a pathologist by training, and he wanted to do something for the cancer in 1947. Okay. And at Boston Children's Hospital, because he was seeing a lot of the kids dying, and they just came in basically, they were all, the ward was considered basically there was a fatal disease at that time, and leukemia was, as a pathologist, he was exactly at the, seeing the outcome of some of these, so at that time, for leukemia, obviously there is no surgery, because the cancer is all over, okay. And now, at the same time, this is an interesting thing as to how the word correlated together. This was an English physician named Lucy Wills in 1928. She was traveling to Bombay and saw that the women who were anemic did not respond just to iron, but responded to paramite, okay? And that was related to folic acid, which was the vitamin, okay, type of thing. And so this was the start of the things. Okay, and so his hypothesis was, Farber's hypothesis was that, okay, well, it makes the cells grow. Can we use that? Obviously, it did not work. It actually was giving fuel to the cells, so it did not work. But at the same time, there was the first Asian Indian who came to do PhD in biochemistry at Harvard. Okay, his name was Yellappa Subbarao. Okay, and he got his PhD from Harvard, but was not given faculty position. So he went to work for pharmaceuticals. There was a pharmaceutical company which has been no longer existed called Ledderly Pharmaceuticals, okay, in New York. And then he was working on anti-folate drugs. So he was able to loan some of these anti-folate drugs to Dr. Farber, called aminopterin, which was the anti-folate. And Dr. Farber designed a trial of aminopterin for B-cell leukemia. And the first patient named Dr. 2-year-old named Robert Sandler achieved a dramatic remission of his B-cell acute lymphatic leukemia with this particular anti-folate drug. And he, obviously the name was kept secret, but Dr. Siddharth Mukherjee, he was able to trace down this man. He was an adult, he got married and had children, and was able to interview him for the series of, Emperor of Melodies. So that's the story as to how three totally unrelated people got together. And again, this is the story that we'll finish up on the positive note that Dr. Farber was ably assisted by Mary Lasker, who we mentioned earlier, and she was helped raise tons of funds, and Dr. Farber and Mary Lasker caused awareness. And this was, they started, there was a 2-year-old who was dying from cancer, and this was started called Jimmy Fund. Okay, and there was a campaign that was started back in early 50s and became a national campaign on radio with baseball stars. Okay, and the baseball players, including Babe Ruth, visited little Jimmy at Boston Children's, and and this is his statue at Boston, in front of Boston Children's. So sorry, but Kevin is not here, but otherwise he can. So this was the start of chemotherapy as we know it, a type of thing, and then later on other drugs came into existence. Okay, so and but before that, people were using. Remember, I think that there one prize was awarded for giving, for treating syphilis, giving malaria vaccine to treat syphilis, to use bacterial. So this was something called Coley's toxin that was tried by Dr. Coley in 1890s, because he saw a patient who had a rip-roaring infection with streptococci called erysipelas. And lo and behold, this person was cured of his sarcoma. And obviously, in retrospect, it could be that the cytokine response took place and the overwhelming immune response. That may be one of the reasons why some people got sick from vaccines, okay, that got overwhelming. And also with the early flu, in the Spanish flu, 1918, the cytokine storm killed a lot of the patients. So that actually was able to kill the cancer cells. So that was called, and he devised something called Coley's toxin with live streptococci. But obviously, that was a pretty risky business to do. So I think we'll stop at this level. and will continue. So I think hopefully we saw that there was some, and later on, the chemotherapy torch was carried on by National Cancer Institute and a small hospital in down south called St. Jude's Hospital. And there was one pioneer oncologist who, single-handedly did multi-drug therapy for leukemias, and their results were spectacular when all the East Coast people were ready to give up on leukemias. As many as 7 or 8 drug combinations were used for leukemia, and they showed some spectacular results with the rise of this chemotherapy and the heydays of chemotherapy that all of us who were in training remember this, giving this drugs to and changing intravenous sites, every 3rd day, and people being sick like dogs and losing weight, losing hair. So that was, some of our experience as residents and interns and fellows with chemotherapy. So I'll stop the screen share, stop the recording now, and stick around for a little while. Stop recording, and I'll send the